Hi, my name is Mijke and I will be talking about learning and innovation in urban yellow mongoose Cynictis penicillata. Learning and innovation has been studied in a variety of taxa, some of which are indicated here, with the intention of establishing exactly what the underlying mechanisms of problem solving are. In these studies, animals are typically presented with either a feeding task or a multi-access puzzle box, or even a trio of tasks that they have to solve. And then their problem-solving abilities are assessed by the definition that problem solving is the ability to use behavior or previously acquired knowledge to overcome an obstacle. Even though a variety of taxa have been considered in problem-solving experiments, studies of animals in urban settings are lacking, despite their abundant opportunity to innovate and their ability to solve problems successfully. For example, New Zealand keas and raccoons have learned to open dustbin lids quite efficiently to feed on food scraps in urban areas. As such, we considered learning and innovation in an urban population of yellow mongoose. Yellow mongoose are small yellow-brown animals approximately 30 centimeters in length that occur in southern Africa. They are opportunistic carnivores that are active during the daytime, and they live in colonies of up to 20 individuals that are often established in urban areas. Our study site was the Meyersdal Nature Area in the south of Johannesburg, South Africa. We had 5 sites in the first location, a residential area interspersed by natural areas with regular human and mongoose encounters. We also had 5 sites in the second location, a natural area fenced off from the residential area where mongoose-human interactions are scarce. And lastly was 1 site in a retirement home where the mongooses frequently visited residential gardens. The aim of our study was to investigate whether urban-dwelling yellow mongoose could learn to solve a task of increasing complexity. We designed a transparent perspex box with a lid, and we presented the mongooses with 4 different stages of increasing complexity, where they had to perform 5, 6, and 9 trials at each of the stages respectively. The mongooses had to learn to open the box in order to receive meat as a food incentive. This design allowed the mongooses to innovate, since they had to use a novel behavior, the lifting of the lid of the box, which is not a behavior that they use in their natural environment, in order to solve a novel problem. We used camera traps to record their interactions with the puzzle box. This first video indicates stage 1, where the lid was completely open and the food could be accessed freely. The second stage, the lid was propped up with a stick, leaving a small gap to access the food. For the third stage, the lid was completely closed. And for the fourth, most complex stage, we placed an object on the lid of the box which had to be removed before the lid could be lifted. The latency to investigate the box, in other words, the time that it took for the mongooses to approach the puzzle box, was considered a measure of neophobia. Using generalized linear mixed models, we found that location and stage were significant predictors of the latency to investigate the puzzle box, but the interaction between them was not significant. The boxes on these graphs indicate the upper and lower quartiles, the horizontal lines indicate the medians, and the error bars indicate the confidence intervals. The latency to investigate the puzzle box was significantly longer in the residential area compared to the natural area, indicating that mongooses in the residential area had a stronger neophobic response. Because mongooses in the residential area come into regular contact with human residents and other disturbances such as motor vehicles, it is possible that this increased interaction contributed to a heightened neophobic response. Next, the latency to investigate the puzzle box was significantly longer in stage 1 compared to all other stages. So the mongooses had an initial neophobic response to the puzzle box during stage 1, which decreased over the subsequent stages. The latency to consume the food, in other words, the latency from first contact with the box to obtaining the food incentive, indicated how long it took the mongooses to solve the problem. Using a linear mixed effect model, we again found that location and stage were significant predictors of the latency to consume with no interaction effect. The mongooses in the residential area took significantly longer to solve the problem compared to those in the retirement home. This slower learning to solve the puzzle box problem could be linked to the differences in the level of disturbance among the locations, leading to differences in the availability of time and energy to devote to solving a problem. The mongooses managed to solve the problem at every stage, but some stages took longer than others to solve. The mongooses were the fastest at solving the problem during stage 1, since they could access the food incentive from the open box. The latency to consume spiked at stage 2, when they had to determine how to lift the lid of the box, but after they learned to open the puzzle box, they were able to open the lid faster during stage 3, since they used the same technique and knowledge acquired from the previous stage. During stage 4, the mongooses had to perform 2 steps in order to open the box, and therefore the latency to consume the food incentive increased again. Looking at the latency to consume over the various trials allowed us to observe their learning on a finer scale. A trial was a single event from the moment a mongoose initiated interaction with a box until it successfully solved the problem. For stage 1, the duration of completing the task stayed relatively constant. However, others such as stage 4 fluctuated, but there was still a general decrease over the various trials, and this decrease potentially indicates learning, or at least becoming more proficient at solving the puzzle box problem. We also looked at the frequency of performing interactive behaviors as indicated here, since it is often considered to be a predictor of learning and problem-solving success. Using a generalized linear mixed model, we found that stage was the only significant predictor of the frequency of interacting with the box when solving the task. There was no location effect, and therefore the interactive behaviors were unrelated to the initial neophobic response. There was very little interaction with the box at the first stage, but the frequency increased with the complexity of the task, especially at the second and fourth stages, since additional interactions were required to solve the problem. So in conclusion, the yellow mongooses in our study showed the ability to learn to solve a novel problem at 4 stages of complexity. In order to solve the puzzle box problem, mongooses had to learn to use a novel motor action to lift the lids, and as such has shown the ability to innovate. They were capable of using the knowledge gained from solving less complex stages to continually solve the problems as the complexity increased. Mongooses in different locations learned at different rates, and therefore their environment had an influence on their problem-solving abilities. Finally, even though we were unable to test a rural population for comparison due to their secretive behavior, we have shown that urban-dwelling mongooses are capable of interacting with novelty in an anthropogenic environment in order to solve a problem. The results from my study opened the doors to investigating yellow mongooses' cognitive abilities even more through current experiments such as reversal learning and divided attention to explore their cognitive flexibility, as well as FID experiments to investigate problem-solving abilities in the presence of humans. This research would not have been possible without the funding provided by the University of the Witwatersrand and the National Research Foundation.